Hello everybody, I'm Molly and I will be helping you through the Biopolymers to Prevent Cancer exercise. It's an amazing activity to explain how seaweed has its properties and how these are being exploited in drug development. More information on how you can complete this experiment can be found in the resource pack on the website. This video is aimed at an activity for secondary school students. If you are looking to complete this with younger students, please head to our website and navigate to the jelly seaweed experiment. If you are completing this with A-level students, we have some extra activities outlined in the A-level Biopolymers to Prevent Cancer section. Let's get into the science. Colorectal cancer is the third most common cancer in men and women worldwide. Almost 55% of cases occur in developed regions of the world. Recent scientific research suggests that a contributing factor could be excess iron in the intestines from a diet rich in red meat. Iron is essential for our diet. For example, red blood cells contain iron and it is crucial for oxygen transport around the body. However, too much iron in the large intestines is believed to cause the formation of tumours. Chemists and cancer scientists at the University of Birmingham are looking at ways to mop up excess iron in the large intestines. The aim is to develop a drug which will bind to iron in the large intestine to prevent the onset of cancer. Researchers are currently looking at a biological polymer, a biopolymer called alginate. Polymers are made up of very large, long molecules with lots of smaller repeating subunits. This biopolymer binds to iron and meets many of the requirements needed for the drug to be active in the large intestines. So let's look at this biological polymer that is being tested to mop up excess iron. Alginates are an example of a structural biopolymer. Structural biopolymers are polymers that are produced by living organisms. They provide plants with their strong stems, crustaceans with their hard shells, and our bodies with flexibility and movement. One of the most interesting things about biopolymers is many of them form gels or viscous solutions in water. Gelatin is perhaps one of the most famous biopolymers used in the food industry, which gives the chewy texture to many sweets. But alginates are also used as a thickening agent to improve the texture of food. Alginate is in fact the most common food additive, also known as E400. Alginates are polymers made up of several hundred sugar monomers. There are two types of monomers, known as high G and high M in alginate. The monomer is made up of blocks of identical or alternating monomers. The strength of the polymer depends on this composition, which varies greatly between different species of seaweed and the growth conditions as well as different parts of the plant. Alginates are potential cancer prevention drugs because they can bind to iron. However, they also bind to calcium very well, which needs to be absorbed by the body for use in teeth and bones. Where there are sections of alginate polymer with lots of G segments, they bind to calcium ions particularly strongly, forming cross-linked egg box regions, and this forms a hard gel-like structure. This effect is used in the food industry. But in order to bind the iron in the large intestines, we do not want the alginate to fill all of its binding sites with calcium. Therefore, researchers at the University of Birmingham are interested in testing alginates that have lots of M segments for preferentially binding iron. The aim of this experiment is to observe the binding of two different alginates with solutions containing calcium and iron ions. To complete this is what you will need. More information can be found on the technician's sheet in the activity resource pack. Okay, so what we're going to need for this experiment is 6 small beakers, a variety of plastic pipettes, a solution of high G alginate, which we're using this brand just of here, and a solution of high M alginate, which is this brand just of here. You're also going to need solutions of calcium chloride and iron chloride, which I will talk you through how to dissolve in a second. It's also best if you use food colouring to add to these, which I will show in a minute to be able to see the solutions clearer. Okay, so now we are going to weigh out 4 grams of the high G and 4 grams of the high M, not adding to the solutions just yet. I'm going to fill these both up with 200 milliliters of deionized water. So we are slowly going to add the high alginate while stirring this. and it should become a very gloopy, honey-like consistency. The slower it is, the easier it will be to dissolve in. You can also very gently heat this. I wouldn't get it too hot because then it'll break down your alginate polysaccharide chains. And I'm going to continue adding the rest of this high M powder to this, and then I will do the same for high G, and I'll see you back in a second. Okay, so it has been about 10 minutes since I mixed these in, and as you can see, they are slowly starting to dissolve in. That's my high G, and that's my high M. As you can see, my powder of high M is a slightly different color to the high G, which is pretty good because you can see which one's which slightly easier. I'm going to leave these, I'm going to put some stirrer bars into them and leave them on a stir plate that's heated at 50 degrees to try and dissolve these down. You could also just leave these for a morning or overnight and they should dissolve into it. Okay, so these are now sitting and stirring. I have poured out 250 milliliters of distilled water because I don't want a litre solution of iron chloride and calcium chloride. Add 2.5 grams of calcium chloride and iron chloride in order to make these 0.1% weight per volume solutions. So once again, as you can see, the calcium chloride has dissolved down to a very, very transparent looking. The water's not really changed colour, however the iron chloride has changed colour because the iron ions. This is helpful for us because it means that we can see the difference in the two solutions again. The same as before with the two different types of alginate. So once these have dissolved and once the high G and high M alginates have dissolved, I'll come back. Okay, so I left these two stirring on the stir plates for about an hour and they finally dissolved down. This is my high M, this is my high G. As you can see, the high M is still darker than the high G. So what we are going to do now is I'm going to add some food colouring to our alginate solutions just so that in the actual experiment we can see them a little bit better. So I'm putting my high M as red and my high G as blue. Perfect. So I believe at this point we're ready to take this into the classroom and do the experiment with the children themselves. Okay, so what we want to do now is we want to get a couple of our pots that we can complete the experiment in and label them. Then I'm going to pour approximately 25 milliliters of calcium chloride into both of these containers. And then what I'm going to do is, using these plastic pipettes, I'm going to suck up 1 to 2 milliliters of the high G and pipette it very slowly so it drops into the calcium chloride solution. Like this. Pretty cool, and it looks like this. So as you can see, a bunch of these little balls have formed, and then I'm going to repeat the same for my high M alginate into the calcium chloride as well. Now, I'm not going to describe the differences between these two because you can fill this out on the worksheet on the website. However, I'm now going to complete the same, but for the iron chloride as well. Add carefully because iron chloride is not great for you. So similar amounts to both. You just got to have enough to see what happens if you drop the solutions in, basically. And then I'm going to do the same for my high G. As you can see, that makes some amazing droplets, and it's making the droplets as soon as it goes into the solution, which is really cool and really clever. And then I'm going to repeat the same for my high M. so now we're going to repeat this again, but we're going to fill these two up with distilled water, and then we're going to drop these in as a negative control. So we see what happens to the alginates when you drop them into solution without any positive ions being in there. This is the high M. So write down all of the things that you can see and try and draw some conclusions of why this would have happened on the worksheets that you can find in the resource pack online. Thank you so much for watching, and I hope you've enjoyed learning about some cool alginate chemistry and how this is being engineered into a colorectal cancer drug. Please check out the other experiments, videos and pupil resources on our webpage, and if you have any questions, feedback or want to request a visit, please use the form in the contact section of our website to get in touch. Thanks so much. Bye.